Welcome to our video that details everything you need to know about DNA replication. Hi everyone, and welcome to our lesson on DNA replication. Now, we've spoken about replication before in science. Specifically, it started in grade 10 when we learned about the cell and the cell cycle and mitosis. And then we learned a lot more detail in grade 11 with mitosis and meiosis. Now, if you recall the cell cycle, most cells are in interphase the longest, okay? And you can see that at the image at the top. And then the actual division phase is the smallest percentage of time that it is in. So replication happens quite a bit every time a cell must create a copy of its DNA before that division phase, before mitosis, and then eventually cytokinesis occurs. So let's talk about this process today. So we know its DNA must be copied. When we talk about making a copy, that is replication. And DNA replication is the process in which DNA is replicated to produce an exact copy of itself, okay? There are no modifications. We're not talking about reproduction when different chromosomes interact yet. Okay, we're just talking about the DNA in a cell without interaction with anything else, and it's going to make an exact copy of itself. Now, of course, there can be errors that can occur, which we will talk about later on in this unit, but the cell does try its best to maintain the full composition of the DNA strand. Now, after the DNA has replicated, each chromosome is made up of 2 chromatids. And we learned this before, but it's always good to recall terminology. So we know that the chromatids are joined together by the centromere. You can see this image on the right side at the top there. And this is what creates that classic X-looking shape of a chromosome. Now, the 2 chromatids will become separated during cell division to form 2 separate chromosomes. And this is why they need to be exact copies of each other. One chromatid, when it gets pulled apart, becomes its own chromosome on one end of the pole, and the other gets pulled apart to the other. And when we're talking about mitosis specifically, we are making exact copies of itself. So DNA replication takes place in the time between cell divisions, as you can see on that cell cycle up there. And this replication process is called semi-conservative because the replicated DNA consists of 1 original strand of DNA. It retains that 1 original strand, and it also keeps that newly replicated, they call it the daughter strand. So you can see in the picture here, the double helix DNA becomes unwound through the replication process. And when it makes exact copies of itself, it retains 1 original strand, or the parent strand, and then it has a daughter strand, which is the newly replicated one. So DNA replication consists of 3 stages, which we will be talking about today. So number 1 is the unwinding stage. This is when the DNA strand is unwound, which allows for replication to occur. Number 2 is elongation. So new DNA strands are made using the original strands as a template. It's elongating. And then number 3, termination. DNA synthesis is completed, and the new DNA molecule, each of them, assembles its own double helix structure. So let's look a little closer at stage 1, which is the unwinding. Without unwinding of the double helix DNA, it would not make the nucleotide bases available to be able to create a new strand. So first we need to uncoil the DNA. And this creates a swivel point, is what they call it. This opens up the DNA molecule for replication. And then there are enzymes that are involved in this initiation phase. It unwinds and unzips the DNA and stabilizes the strands so that the copying can begin. So one example of an enzyme in this process is DNA helicase, which actually unwinds that double helix to begin this replication, to begin the replication process during stage 1 of unwinding. Okay, you can see as it becomes unwound with DNA helicase, we could see that there's a replication fork that occurs. This is where it's splitting and splitting and splitting upwards. Okay, and that can create temporary breaks, which is called a swivel point. So stage 2 in the replication process is elongation. And we end up with the unwound DNA strands that is going to be used as a template. Now, these original strands, again, are called the parent strands, the ones that just unwound. And new DNA is formed by adding free nucleotides. Free nucleotides are actually free-floating around the nucleus, and they can be picked up during this process. And with the enzyme DNA polymerase, that will catalyze the bonding along this DNA strand with its complementary base pairs to form the new daughter strands. Okay, so the new strand is the daughter strand. You can see those in the image as the orange ones that are being formed along the parent strand. And what's important, again, is to reiterate that the DNA and RNA strands elongate in the direction of 5' to 3'. So if you look at the left side there, for example, you can see the 5' end of that daughter strand of that misstrand orange one is at the bottom, and it's going to elongate towards the replication fork. Okay, and that's where the nucleotide bases are being added. In contrast, you could see the other daughter strand, right? It's forming complementary base pairs with the parent strand of the other side, the anti-parallel side. So it's going to be replicating still in the same direction of 5' to 3'. However, you can see it starts at the replication fork. It's not moving towards it and adding free nucleotides. It actually starts at the replication fork at the 5' ends, and it ends up adding new nucleotide bases along it. And you can see the 3' end is at the bottom there. They always run anti-parallel with the parent strand as it forms new ones, just as regular DNA structure dictates. So here's a closer look at the 5' to 3' ends and the anti-parallel configuration. Again, they're parallel, however, they run in opposite directions. And the direction is dictated by the way that the new nucleotides are bonded with each other with the phosphodiester bonds. Okay, and the last stage, we're almost there. We need to end this process. So when DNA synthesis is completed, the process is said to be terminated. Each new DNA molecule contains one strand of original DNA, that parent strand, and one strand of newly synthesized DNA, which is the daughter strand, which will be it should be the exact copy of its complementary base. So it will be exactly the same as the other strand that pulled apart during the initial process. The two new strands of DNA now coil back up into a helix as a chromosome, and the newly formed chromosome will later split into 2 chromatids during cell division. Okay, we're not there yet, but this is a precursor to the division process, which is why we like to mention it, since it is what we're working towards there. Okay, so you can see termination occurs and the recoiling occurs at this point. Let's just reiterate nucleotide matching, as it is very important. During the DNA replication, new nucleotides are added at the region called the replication fork. I pointed that out before, but it's exactly where the 2 original strands start splitting apart there. That's called the replication fork. And free nucleotides are matched with their corresponding template bases, as we've talked about many times, A with T, G with C. And we follow Chargaff's rule. We mentioned Chargaff yesterday, when he realized that there are always equal amounts of purines and pyrimidines. And this is the rule that he came up with the A and the T and the G with the C.